My name's Melissa McAlister, and I'm going to be talking to you about Watershed Watch's project to build local capacity for the detection of sewage-related water quality threats in eastern Kentucky. Our organization supports water quality monitoring across the state by volunteer monitors who are interested in helping protect water quality. Watershed Watch coordinates 3 different sampling events each year during which we analyze field chemistry but also collect grab samples to be analyzed at labs for E. coli that helps us better understand sewage threats as well as other wastewater sources. And we use the labs identified by black dots on the map where you can see that we have big gaps that cause volunteers to have to drive long distances. We attempted to address this somewhat in eastern Kentucky with labs denoted by the 2 yellow dots on the map. So we were trying to address limitations to volunteer monitoring by enhancing convenience of the labs, but also addressing costs, since lab analysis for E. coli typically cost around $25 a sample. So we were piloting a less expensive alternative called ColaGlow. The 2 project goals were to establish the 2 mini labs in eastern Kentucky and also compare the trial method of ColaGlow versus the established IDEX method for E. coli analysis through 2 focus studies, and our hopeful outcomes were to have longer-term partnerships with these mini labs for continued use for Watershed Watch, and also have an option to use ColaGlow as a simpler, less expensive analytical technique. This map shows the project areas in green where the focus studies were in those watersheds, and then the blue highlighted labs at Lilly Cornett and Glady Nature Center. We did training workshops in the fall of 2021 and conducted sampling between December and February. And then we analyzed the data to compare the 2 E. coli analysis methods versus Watershed Watch's grading system, and also asked the volunteers for feedback on using the 2 methods. In comparing the 2 methods against the Watershed Watch grading system, we found that the 2 methods were more comparable at higher concentrations, which produce worse grades, and less comparable at lower concentrations with higher grade levels. Frequently where the 2 results differed, we did see that ColaGlow analysis often produced higher concentration readings than IDEX. Statistical analyses did show a strong relationship between the 2 methods, which was encouraging. In conclusion, the 2 new labs that we set up in eastern Kentucky were used for sample analysis during the 2022 sampling season. We are working on more streamlined logistics for those labs for 2023. And regarding the method comparison, we determined that ColaGlow is an effective alternative for analyzing E. coli, although we'd like to do a little additional study on the role of sample dilutions and skewing some of the results. The participant feedback was also supportive, and so we presented ColaGlow as a feasible alternative for use in remote locations across the state.